some information that you already see on the website if you have accessed that and were curious. Bachelor of Science in Molecular Biology and Bioinformatics, which is the same field that Matthew Cherhutty has studied in. From the University of Wisconsin-Parkside, PhD in Cell and Developmental Biology from Harvard University. Developed the Biological Research Program for the Institute for Research. Currently research biologist at Answers in Genesis. He is the author of Replacing Darwin, which he talked about last night. New Origin of Species. Replacing Darwin Made Simple. And that sounds like it's about on my level. And Traced Human DNA, big surprise. He talked about all three of those yesterday. I hope you mention them again, because we've got a few people that are new today that haven't been here previously. So without any further ado,
thank you very much. Can you hear me? Yep, there. Okay, we're on. This will not go as long as it did last night, because I was the last one and we had a little extra time, so I just kept going. Perhaps not as long as I thought I was going to keep going, but there we go. I'll change one more thing here. Get rid of, if you're watching on the video screen, I'm just changing some things for our display right here. There we go. And I'll come back to the display where you can interact with me here in a moment for Q&A and such, and try to make sure I repeat the questions. Now we're going to go a little bit off the beaten path here. If you've been in creation science a while, so I want to set a bigger stage here for us as to my philosophy of the role of the apologist and why this particular topic and how I go about doing it, and the big takeaway it relates to the overall picture. So just to review from last night, and I'll go through some of the specifics here in a moment of what we discussed as it relates to species last night. But the big picture that I wanted you all to take away if you're with us then was some of the questions, that there are more questions surrounding the origin of species than we commonly assume. And the point of today's lecture will be to show that there are many more answers than we commonly assume. And both of those can be true at the same time. But the type of answer that we're going to look at is perhaps where we go off the beaten path, and is not usually what's discussed in creation science circles, but is apologetically, I think, very powerful and relevant. So just to set the context, the larger context, I want to ask and answer three questions very briefly before we get into the meat of what we're going to do. And sort of dovetails with what we just discussed in the hour before lunch. What will it take to move the needle? So if you look at surveys of the general populace, if you look at surveys of the scientific community, there's a fairly big discrepancy. The general public, about a third of the general public will generally ascribe to something sounding like creationism, much to the chagrin of the professional scientific community. And if you look at the survey of members of the American Association of the Advancement for Science, let's say, like, okay, this is still working. Hear my sound different. There's almost 99% of them would say they hold to evolution. And hardly any would give some sort of creationist answer. And if they're the ones in charge, that's what the public school kids hear year after year. This is what's taught to the next generation consistently. So what would it take to move that needle? And so the three questions I want to ask real fast are, what does Scripture say as the problem? Make a brief comment about what's been the history of this exchange, some of my own experience as well. And then some of the barriers, what sort of barriers exist to moving it right now. So in terms of my scriptural framework for thinking about how you counteract this, because that's my job basically, and I think a lot about this. I always go back to Romans 1, which says the things of God are clearly seen in nature. Actually, if we go back to verse 18, that's verse 20. Verse 18 says, I don't have it up here on the screen. It talks about the unbelievers suppressing the truth in unrighteousness. And I think that's the key phrase. And perhaps a useful anecdote here would be Dave Menton, Lutheran, who passed away, and taught at the Washington University School of Medicine for 33 years. He said he would ask his unbelieving colleagues, actually, let me back up a second. He said he'd ask his believing friends if there was ever times that they questioned their faith, there were times of doubt, or if it was always just very strong belief in the Lord. And he says most Christians that he talked to would answer that, yeah, there's times of waxing and waning with their faith, and times of doubt and times of trial, times where they feel strong in their faith, these sorts of things. There's variability. And then he said he would ask his unbelieving colleagues if there was ever a time that they doubted their unbelief, lay awake in bed at night and wonder what they hold to is actually true. And he said, to a man, all of them said no. They never doubted. And I'm inclined to believe them because Romans 1 says this isn't something passive. The unbeliever doesn't just kind of wander through life and, well, maybe I believe, maybe I don't. They make an active decision. No, I don't believe this. I know the truth about God, and they put their foot down and say, no, I don't believe it. They suppress the truth in unrighteousness. So what then is the job of the believer, of the apologist, if this is what's going on out there in the world? I'm speaking to Christians now. What I see as my job, and perhaps all of our job, if you're a believer, is to remove the excuses for suppressing the truth. The Holy Spirit needs to convict them of sin and of the gospel. They're making excuses for it. In this case, in the Western world, evolution is a common excuse to say, sorry, I don't believe in God. Richard Dawkins knows there's a God, but he suppresses the truth in unrighteousness. And so the job of the apologist, whether professional or lay, is to remove those excuses and say, sorry, let's get back to the main issue, which is you don't want to deal with God and your sin. That's the big picture philosophy I take. That'll be relevant to what we discuss here in a moment. In terms of how this is played out, I want to add to the history that was mentioned the previous hour. Duane Gish was probably the foremost debater in all of creation science. He was with the Institute for Creation Research. I think he did about 300 debates, probably would have kept going. But it was the evolutionists who said, we don't want to do it anymore. Stephen Jay Gould said that the best the evolutionists could do in such scenarios was a tie, sort of a backhanded compliment to the ability of creation scientists to win debates. That the best the evolutionists could do was a tie. He acknowledged that. You had something to say.
Yeah, about Duane Gish. When I was an undergraduate at University of Minnesota in the 1970s, he debated the head of the biology department, University of Minnesota. The debate was held in the largest auditorium in campus. A couple thousand people were there. And he beat the pants off that biology professor. He was not prepared, you could tell. And Duane Gish had all this evidence, and that opened up a lot of eyes.
Yeah. So for those watching, anecdote about a Duane Gish debate back in the 70s, and tried to allude to some of this history last night, where the 70s were sort of the heyday, and the evolutionists will have admitted in print after the fact we weren't prepared for what the creationists brought. That progressed to sort of a backhanded, even when we were prepared, they're just kind of clever, and this isn't the right format for it, which again is a sort of a tacit admission that they were still losing. And it became very difficult to find opponents. So I've done about 3 debates. It's still difficult to find someone who wants to contest this. The 3 I've done are with Denisovans, who's a theistic evolutionist on the campus of Southeastern Seminary, in which his goal was not really to debate any of the facts, but to present a we're believers too face. That's their main philosophy. The second one was online with Herman Mays, who's a PhD biologist at Marshall University in West Virginia. I found out, or was reminded after the fact, that he had written a blog post several years prior titled something like In Defense of Ridicule, in which his point was it's long past time to give the creation movement any oxygen. We should not even platform them. We should simply mercilessly make fun of them and ridicule them as a strategy. And if you watch the debate, we have it online. You can go to Ancestry and Genesis webpage to look it up. I had a 12,000 word summary after the fact, writing up about it. We basically talked past each other because he didn't hardly but skim the book we were supposed to debate, Replacing Darwin, which is what you do if your philosophy is, I'm just going to make fun of the opposition, not even try to engage it. I just had an unplanned debate about 3 weeks ago, June 2nd, with a guy who came into a call-in interview show. Daniel Cardinal is the guy's name. He's a biology professor at Rutgers who had reviewed this new book I wrote, Trace, and he had his points prepared. There was no organization of it. Again, anyone who wanted to call in could, and so he came on and tried to debate me for an hour. And you can decide for yourself. I've got a link on my blog for finding it. I don't think he was prepared for the answer. I put in print that my explanation for why so many professional scientists disagree with creation science is because they're never taught an alternative view. The counter argument to that is that you have people like Daniel Cardinal who said, I've read your book, I've reviewed it. What you'll see in that debate is he couldn't articulate hardly any of my points. And you can't make this up. Truth is stranger than fiction. I'm answering his question. You can watch this in the video. He's literally typing something else on the computer, moving his coffee mug around, refusing to listen to anything I'm saying. That's the state of affairs when it comes to debates. The leading voices trying to take on creation science either don't want to acknowledge it. And let me give one more anecdote. When Replacing Darwin came out, we sent a copy to Richard Dawkins, to Jerry Coyne, to P.Z. Myers, in hopes that they would tell their students not to read it. And you have this sort of reverse psychology effect, be rebellious, read the forbidden book. And they're wiser than that. They didn't say anything. I know P.Z. Myers got it because he just opened his mouth now, because someone had criticized this new book, Traced. And he announced, well, Jeanson sent me this book back several years ago, and I put it on the shelf and didn't say anything about it. I'm like, okay, you admit you got it, but he knows better, doesn't want to say anything because of this. There's this reverse psychology effect. So the question in my mind is, given the pattern, they've done debates, they admit that we can do it so well, and finally say we're going to stop because the best we can do is a tie. A tie doesn't help evolution. Next phase, you've got some saying there's no point debating the facts, let's just make fun of them mercilessly. Then you've got a guy just a few weeks ago, I'm going to try to debate him, and while he's answering my question, I'm going to ignore it completely. Why is the opposition to creation science so insistent on shutting down debate? I think it's because they feel threatened by it. I mean, you don't have to agree with me. That's just a question I think to chew on. Why are they so insistent on shutting it down? So what does it take to move the needle? What barriers exist? This is the last question I want to answer. What barriers exist to moving the needle? We have newspaper reporters, often hostile ones, come to me and say, well, you're a creation scientist. Yes, you know that 97, 99% of the scientific community disagrees with you. Yes. So what is it? Do you reject science entirely? Or is there this vast conspiracy to suppress the actual facts? Poisonous door number one or poisonous door number two? Which, of course, it's a trap question. I say neither of those. Option number three is a self-fulfilling prophecy. In the United States, you have basically forbidden the teaching of creation science in the public schools, which is where the vast majority of students go through. Pretty much every professional scientist, almost guaranteed, has gone through the public school system. They're never forced to read a creation technical paper. They go through college, they go through graduate school, never exposed to the other side. So you put evolution in, do you expect anything but evolution to come out? Why do so many people reject creation science? They don't even know it exists. They're not even aware of creation science. The evolutionists control the funding streams. I was on that. I read the evolutionary blogs, read what the other side says. And it's kind of funny. One day, one of the guys sort of is reflecting and saying, we criticize the creation scientists for not doing science, but we never give them any funding for it. So maybe we shouldn't do that. Or maybe we shouldn't expect them to do science if we work so hard to cut off all their funding sources. And that point was kind of just dropped and they moved on. But it's something I've thought about, because how do you move the needle? The opponents of creation science at every single level, funding, education, have put up barriers in place, even to the level of debates, to keep people from hearing the other side. So my philosophy is, going back to the scriptures, they're suppressing the truth. And I think I've just given you several more examples of that, how they're suppressing the truth. No funding, no exposure, none of these sorts of things. How do you push back against that? And what I take as philosophy right now is exposure, exposure, exposure. The opponents of creation science are so concerned about students being exposed to this, so that they put laws in place so they can't hear it. They shut down any sort of debate in public. Why not try to make it as public as possible? So what I want to expose you to now, in the minutes that remain, are, again, or what I should say is a single point, a long extended example of there are more answers to the origin of species than we commonly appreciate. And just to tee up where we were last night, when we talked about the mystery of species, We said if you look at species, I tried to derive for us. So again, if you weren't here, I'll rederive it again for you very quickly. We tried to derive the mystery of species. I said if you just look at species, there isn't much mysterious about them. They reproduce more of themselves. But if you look below the Earth's surface, there's obviously something different about species that once existed as compared to today. So what happened? We started with the zebra as an example, just asking and answering some basic questions. Where do they live? They live in Africa. Why do they live there in Africa? Because they've survived there. How do they survive? Well, stripes seem to play some sort of role. Zebras don't exist in every instance of the savanna. Let me back up a second. The point I wanted to make here was zebra stripes are designed for a purpose. They help the zebra survive in Africa, in the savannas of Africa. They don't exist in every instance of savannas in the globe. And to make a long story short, what we derived was it appears that zebras exist in Africa because they've migrated there. It's not just one species, but several species of zebra that exist in Africa. And the most parsimonious or statistically plausible explanation for the existence of 3 zebra species in Africa and nowhere else was that perhaps one original ancestor migrated in and then diversified into the 3 species that we see today. So a geographical argument for common ancestry. We talked about the various breeds of horses that exist in the world today. But there's more variety among the breeds than among the various species that we see. We know by definition that these various breeds arose from a common ancestor because humans were responsible for it. So if these breeds came from a common ancestor, why not these species that have less variety? And the mystery, so what I'm driving towards here and what we derived last night, the mystery of species is where does common ancestry stop? Where do you draw the line? That these individuals on the screen all come from a common ancestor. Zebras and whales have a common ancestor. That's the big mystery. Answering that question is harder than it looks. The path forward we began to sketch was this concept of hypothesis testing, the basic way science is done, and also one of the main criticisms of creation science from the evolutionary community. And I'll give you a quote here again in a moment, but the long-standing criticism from about 1980 onward is that creation science isn't science. You've probably heard just the lay version of this. Creation scientists don't do science. Creation science is an oxymoron. It's actually religion disguised as science. It's pseudoscience. We said this is the form that a scientific hypothesis should take, and you should be able to then go out in the future and do some sort of experiments. We walked through some of the main textbook arguments for universal common ancestry, that all species are ultimately related back to a set of common ancestors. We looked at their underlying structures. This is one of the main arguments that the evolutionists make. We saw that if you think about, or if you try to derive the principles that God may have implemented when he created the kinds in Genesis 1, we took some inspiration from how humans design things because we're made in his image. There's something about us that reflects something about God himself. So how do humans design things and what patterns do they design things? They design things in, for lack of a better term, a homologous way. There's underlying shared structures among these various vehicles. I don't have time to rederive it all. I apologize again for those of you who are here for the first time. The point being, though, this observation right here of a similar underlying structure across these various species is as much a prediction of the creation science model as it is of evolution. That's effectively then a tie in terms of adjudicating which model is correct. We talked about the possibility of transitional forms, and I short-circuited this debate not by going through the paleontological evidence, I'm not a paleontologist, but simply saying humans produce in design what we might otherwise call a transitional form. Things that blend the features, in this case, an amphibious assault vehicle blends the features of tanks and ships. Not a perfect ship, not a perfect tank, but perfect for the transitional environment between the two. Again, this is all just review from last night. The main evidences for evolution, just using these 2 arguments as an example, are also predicted by young Earth creation. None of the major lines of evidence for evolution adjudicate between creation and evolution, and so therefore cannot be used as evidence for evolution. We talked about the possible way to resolve that by talking about so-called bad design or poor design. It'd be a great way to, in theory, refute the idea of God designing things, but this is an argument from ignorance or from silence, and as experiments fill in that ignorance, all these supposed examples of bad design have basically gone away. So that was a very fast-forwarded review of where we were last night. The point of last night was to, again, show that there are more questions surrounding the origin of species than we commonly assume. And the point for this afternoon is to show that there are more answers to these questions than we commonly assume, but perhaps not the ones we typically talk about or think about. I showed you last night some of, basically, in a nutshell, just a few slides, Michael Behe's work, Darwin's Black Box, taking Darwin's own statements about how you would test evolution and showing that modern genetics effectively refutes evolution, effectively refutes universal common ancestry as an explanation for all of life. But we never quite fully answered the question, what is the real explanation for the origin of species? Let's say evolution is wrong. You can't connect everything back to an original single-celled form billions of years ago. Well, could you connect some species back to a common ancestor? Could you connect whales and zebras? Where do you draw the line? We're still back at that question. And I'm suggesting that hypothesis testing is the way forward. And I want to do that now. So now we're on to today's talk, finally. What I want to do now is use the question of human origins as a case study for this, as, hey, I think this works for this particular question. So I'm optimistic it'll work for the rest of the species. So here's the quote that I said I would give you earlier. Why am I talking about hypothesis testing? Because number one, the evolutionists have been arguing against creation science along these very lines for 40 years. I'm giving you a more recent quote, just because it's concise and for sake of time. So now we've got whole chapters in evolutionary textbooks dedicated to dealing with creation science, trying to refute creation science. And one of the main arguments is that the most important feature of scientific hypotheses is that they are testable. Again, using gravity as an example, gravity predicts that if I release my grip on this water bottle, it'll fall. Of course it does, but if it started levitating, in theory, that would have been a refutation of this idea of gravity. Evolutionists want creation scientists to come up with some sort of statements that future experiments could reveal to be true or false. So I said human origins is the main question we're going to focus on. In the evolutionary view, humans shared a common ancestor with chimpanzees 6 to 13 million years ago. The human branch, use my laser pointer here, came off the chimpanzee, or what led to the chimpanzee branch back here. The human lineage went through a series of intermediate forms. Neanderthals being one of those forms, they would say, before about 200,000 years ago. Anatomically modern humans arose, or 200 to 300,000 years ago. The reason I bring this up is, in light of what we discussed last night, is the refutation of evolution by Darwin's own standard enough to reject this view? So just to review from last night, Darwin said, if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Can you build humans from an ape-like ancestor step by tiny step? Are there any structures that you can't do? So just to perhaps make it more provocative, last night I talked about the example of sexual reproduction, male and female. You have to have both for the system to work, otherwise it breaks down. So here's an example where you need an evolutionary miracle to explain it. Well, by the time we get to this stage of evolution, we already have sexually reproducing species. That's not something that needs to be done to get to humans. So what if we're at this stage? Can you evolve anatomically modern humans by evolutionary processes? Is this enough of a refutation? And again, part of the reason I'm bringing this up is Michael Behe himself, who's popularized this argument, part of the intelligent design community. He is okay with common ancestry of humans and chimpanzees. He has no problem saying that we're basically evolved apes. He just doesn't think that mutations in natural selection are the mechanism by which that occurs. But in terms of common ancestry, he's fine with it. So how do we figure out the sticky question of where common ancestry stops? And again, this is perhaps the most personally relevant, pressing question for all of us because we're humans. And how do we effectively say yay or nay that chimpanzees are in our family tree? Is this enough? Again, the point I'm going to try to make is that hypothesis testing is the way forward on this particular question. Now, if I can answer this particular question satisfactorily, I want to say that there's a broader application of this to the rest of the species. One of the counter or contrasting explanations for the origin of humans, of course, is the biblical model, the young earth model. That God creates supernaturally Adam and Eve 6,000 years ago. They obey the command to be fruitful and multiply. They have many offspring, many descendants. They go wicked, of course, after the fall, or Adam and Eve sin at the fall, and all their offspring then are wicked. The whole world goes wicked. God decides to destroy all but 8 of them in the flood. Noah and his wife, his 3 sons and their wives, about 4,500 years ago. Humanity restarts in the Middle East somewhere, and then instead of spreading out like God commands, they build a tower in a city, and so God confuses their languages to force them to spread out, and then 4,000 years later, here we are. How would we know whether or not this explanation for human origins is true scientifically? How would you know that ancestry for humanity stops right here? Do not continue further in the past to connect to some sort of great ape ancestor. The evolutionary standard is testable predictions. Statements about the future that future experiments could reveal to be true or false. And so I'm going to give you the answer, sort of the point of what I'm going to show you, and come back to this point over and over again so we can follow it. I will say that the young earth creation model is true because this model has produced a historical progression of discoveries in which successive testable young earth creation predictions keep working. That's a mouthful of a sentence, but it has this idea that how do we know this model is true? Because it keeps predicting things that work. That's the ultimate standard of science. It's all pragmatic. Does it work or not? So in the next few minutes, what do I have here? Half an hour. Next 30 minutes, I want to walk you through basically a history lesson. 10 years worth of experiments in human genetics by young earth creationists, in which there are predictions made, fulfilled, the evolutionists come in and criticize. More predictions are made, they're fulfilled, evolutionists come in and criticize, and this cycle keeps repeating. And that is the gold standard that evolutionists have insisted on for 40 years. So this is a big deal, is my point. Hypothesis testing, I'm arguing, is the way forward. It's working very well for the question of human origins, which I think then is a good marker of things to come, an optimistic sign of things to come. I'm going to walk through a lot of what could be technical material. I'm going to keep it simple. The point here is not that you walk away and can articulate every single detail of what I'm discussing here. What I want to show you, again, is just a history of discoveries. This is a history lesson, not fundamentally a molecular biology or genetics lesson. But just so that we can all be on the same page, I want to describe for you the 3 main genetic compartments when we talk about human DNA. You may not even know there were 3 genetic compartments for human DNA. Well, here they are. We typically think about what's called autosomal DNA, the stuff that gets all the attention. If you take an Ancestry.com test, this is the type of DNA that they're testing. It's the DNA you get from both parents. It's 99% of our DNA, and because it's 99%, we hardly talk about anything else. The 2 other types of DNA come from only one parent. Mitochondrial DNA, as best as we can observe, comes only from mom. Males and females have it. I have it from my mom. My 4 children have their mitochondrial DNA from my wife. Y chromosome DNA comes only through dad. Unlike mitochondrial DNA, only males have it. Females are XX, males are XY. I have my dad's Y chromosome. It's inherited that way. And those different types of inheritance aren't just defining features of these various compartments, but they play a big role in how we understand their origins. I got started working on species questions, human questions, and Matthew's referred to this in his talks, working with mitochondrial DNA, the stuff that's inherited through mom. Now, again, I'm going to try to focus and tell you just the human side of the story, though some of these studies involve many more species. This type of DNA undergoes mutations, copying errors, every so many generations. I'll tell you about the exact rate here in a moment. When I first came to the Institute for Creation Research, just to back up the story even more, 2009, the first thing I was tasked with was developing a biology research program. So the first thing I did was find the questions that need to be answered and then find the tools. And it just so happened, again, Matthew's referred to this, mitochondrial DNA is short. In humans, only about 16,000, 17,000 letters long. So it's fairly cheap and easy to sequence. Fairly cheap and easy to sequence for other species as well. And so at that time, 2009, there were almost 3,000 species that had mitochondrial DNA sequences. This is a first major paper I published in 2013 because it was a several-year study in which the primary focus of this paper was trying to develop a testable predictive model for explaining differences between kinds. I'm not going to spend time talking about that because the narrative I do want to tell you relates to something I put at the tail end of this paper. Again, it's fairly cheap and easy to find a species, get its mitochondrial DNA, sequence it, publish it. If you want to measure the rate at which it changes, you've got to get multiple pairs of mother offspring, mother-daughter, multiple times over, many more individuals. Get the DNA of mom, get the DNA of daughter, count the number of differences. That's what you need to do to get the mutation rate. So even though there were 2,700 species that had a published mitochondrial DNA sequence, okay, tiger, lion, bovid, so on, there were only a handful of species in which this sort of mutation rate measurement had been done. So here on the screen, roundworms, Canorhabditis is the genus name, fruit flies, Drosophila is the genus name, water fleas, Daphnia is the genus name, and then of course humans. And just to tell you that the swath of life that is represented here, humans, as Matthew talked about earlier or last night, are classified as vertebrates, chordates. Both the water flea and the fruit fly are classified as arthropod. That's a whole separate phylum. Think of one of many types of invertebrates. And this nematoda is a whole separate animal phylum. So there's 4 species, but 3 phyla, pretty significant swath of life represented here. And what was exciting about this is that in each case, the rate of mitochondrial mutation the rate at which mitochondrial DNA mutates had been measured. And in each case, the answer was the same. So the numbers here are probably hard to read, but you can see the big picture implication. In every one of these 4 graphs, and this is just a screenshot from the paper, the red bar represents the predicted number of mutations under the evolutionary model. So it's tall. So here I've taken from the evolutionary literature the time of origin for roundworms, for these fruit fly species, for water fleas. So for roundworms, the evolutionary literature said they had diverged 18 million years ago. Fruit flies, 20 million years ago. These water flea species, 7.6 million years ago. For humans, 180,000 years ago. Actually, I had to revise that down to 50,000 because I was only looking at non-Africans, but that's another point from the day. In every case, you can see very tall red bars, because if you're mutating for millions of years and all sorts of generations, you're going to accumulate tons of DNA differences, more than the actual sequence itself. So in this case, this vertical axis is this bar almost reaches up to 2.5 million. Effectively, that means these guys would have mutated their entire sequence, their entire mitochondrial genome multiple times over. How could they survive that? The green bar represents the measured number of mitochondrial DNA differences between the various roundworm species, or between the various fruit fly species, or within the water flea species, or here among various humans, non-African humans. And then blue is the predicted number of differences after 10,000 years. I started with 10,000 years, sort of as an upper limit. I revised these down to 6,000 in subsequent papers. Again, the big picture image here, you can see that the green bar in every case, you can barely see because it's so short. The blue bar, young Earth prediction, again, is just as short as this one is over here. And red is a huge over prediction. So in every case, you have a strong argument against evolution and for creation. Confirmation of creation. There appears to be this mitochondrial DNA clock that marks off the passage of time. In humans, it's been measured to be about 1 mutation every 5 to 10 generations. And so in 6,000 years, you can explain the diversity that we see in non-Africans. Okay, this isn't necessarily a prediction. This is taking existing data and seeing which model, the evolutionary one, the time scale, makes more sense of this, or does that make more sense of the data, or does the young Earth model make sense of it? That's the initial observation. And again, strong arguments because it's consistent across multiple phyla. But the bigger question, of course, is what predictions does this finding make? Once you have a consistent pattern that leads to predictions, in fact, to fast forward, I think I put predictions in this paper. If not, I put them explicitly in the 2017 book, Replacing Darwin. I said, okay, there's a consistent pattern here across invertebrates. this tells me that we can make a prediction, and I put it in print, a formula. Here's how we can predict the rate of change for the grasshoppers outside, or the katydids, or the earwigs, name some of the species. There's now, the young earth model can predict the rate of change. In this case, so I mentioned that I had to revise this number because I was only looking at non-African peoples. The reason I was only looking there was the published rates of mitochondrial mutation in humans were only measured in non-Africans. So I compared non-African data to non-African data. And then this allowed me to say, I'll make a prediction for the African data. And this is what I've been challenging my evolutionary colleagues with. You want to find fault with what I'm doing, make your own prediction. And we can do this head to head. So science is to be done by their own standards. Okay, there's what I just said there on the screen. Some of the objections that were raised at this time and have been raised since then deal with how these experiments were conducted. So for example, I mentioned here that you're looking at mothers and daughters. You get mom's DNA, you get daughter's DNA, how many differences exist? And so you get about 5 mother-daughter pairs, 5 to 10 mother-daughter pairs, and you'll find a difference. That's a measurement basically in the present. And the evolutionists of all people have been saying, well, how do you know that's been consistent in the past? Which of course, if you've been in creation science, you know that in the field of geology, this whole idea that the present is the key to the past is the foundation for all of millions of years. They always assume present is the key to the past. Present rates dictate the past. Now that it says the opposite into the field of genetics, now they want to question this assumption. It's still a fair question to raise. How do we know that this has been consistent in the past? And the other objection, okay, I have a slide here too, or to put it this way, another form of that questioning of uniformitarianism is, well, how do you know natural selection didn't play a role? Especially when you look at the back up a few slides here. If you look at the magnitude of how many mutations would have occurred, they'll say, well, surely they wouldn't have mutated in every single position. Natural selection would have eliminated those that were lethal. So it's still a fair question to raise. My response to them would be, we'll come up with a model based on natural selection that makes predictions. And if you don't make predictions, you're doing pseudoscience by your own standard. Two can play this game. So that's one objection that these original studies didn't necessarily explicitly address. The other objection had to do with the fact that I was only looking at DNA from living species, living individuals, and not from fossils. We talked about this briefly last night. This is objection that's raised. And the reason it's raised is this green bar is the number of differences that I show right here is based on DNA from living people. If you throw in Neanderthal DNA, this green bar goes up, which brings it closer to the red bar and makes it higher than the blue bar. It basically takes what's agreement and makes it disagreement. So that's part of the reason the evolutionary community raised this question. Next paper I published 2 years later, talking about the mitochondrial DNA clocks across various species, rates of change. There's something else, again, that most of the paper deals with animals. I want to just draw out for you the human data, because that's the point of this talk, is to show you the human narrative, how it's progressed with time, predictions, fulfillments, just like the evolutionists have demanded. One of the things that came up in this paper, based on mitochondrial DNA again, was some details about the human mitochondrial tree. So the fact that this mitochondrial DNA mutates like in a clock-like manner and adds the amount of diversity every so many generations means you can basically reconstruct human history. This would, for sake of time, I won't fully derive this, but basically you can get a family tree based on mitochondrial DNA and look something like this. This is based on the DNA from about 300 to 400 individuals. You can't actually read the names, but those letters correspond to one person right here, another person right there, another person here, you know, there's several hundred. I don't have it zoomed in here because I want you to see the big picture. In this case, the tips of the branches, here's a tip, here's a tip, here's a tip, here's a tip, here's a tip. That all represents the present. And so to go backwards in time, you have to move away from the tips. So in a sense, you've got a pseudo bullseye right here, and time moves inwards towards the center of the bullseye. So that would be somewhere close to the beginning. That's what I'm arguing. And what I want you to see here is that near the beginning, there's three major nodes where a whole bunch of branches come together. There's another node right here for reasons I don't have time to get into. That's just an artifact of what's going on. At the very base, even the evolutionists acknowledge that there's three major nodes. They would call these L, N, and N. They've got letters for them. This is not something that's even controversial in the evolutionary literature. Why am I bringing this up? Number one, this is a family tree based on mitochondrial DNA. Number two, this family tree has three major nodes near its beginning. And the third point is, when you think about biblical history, the biblical history applied to mitochondrial DNA, you find a very intriguing correlation. I mentioned, so here's where the biology of the inheritance plays a role that I mentioned just a few slides ago. Mitochondrial DNA is inherited through mom. So the three boys, Shem, Ham, and Japheth, get their mitochondrial DNA from their mother, but then it stops. They don't pass it on because they're males. We all get our mitochondrial DNA from their wives. And the scripture is silent on the identity and ancestry of these wives. Well, we know they all go back to Eve, but it doesn't say whether or not these ladies were daughters of Noah and Mrs. Noah, whether they were sisters, or what sort of relationship they had. Because they're not mentioned as daughters, this is an argument from silence, I realize, but I'm less inclined to think they were daughters of Noah and Mrs. Noah. I think they were not. Whether or not they were sisters, I don't know. They could have, not had a common ancestor until you go back to Eve. The point is, you have in theory three female beginning points around the time of the flood, and you have in this mitochondrial DNA tree three major nodes. This would seem to suggest then that the rate of mutation, which I'm assuming right now, 5 mutations per generation, has been consistent through the past because it fits the biblical history. Indirect argument in favor of the rate of change being constant. There was another thing that I brought out in this paper. I threw in some fossil sequences, the Denisovans and the Neanderthals. And here's the logic that I tried to use in the paper. So I've changed the diagram now. We've still got a mitochondrial DNA-based tree, but instead of looking at 300, 400 individuals, I'm going to look at 53 modern individuals plus 3 fossil sequences. So those 3 nodes now are way back here. You can barely see them. These are the long branches here that are African. And lo and behold, the fossil sequences branch off the African part of the tree. Why does that matter? So here you can see how long the branches are. That's why it would raise that bar a little bit. Anyway, let me back up for a moment and make a point here. So one of the predictions that I made, put in print, I said if we measure the rate of change for this part of the tree, for this part of the tree, we get about 1 mutation every 5 to 10 generations. No one's measured it in this part of the tree, the African part of the tree. Here the branches are longer. The predictions I made is that the rate of change in the African branches would be 2 or 3 times faster than what's been measured. The evolutionists, you can kind of infer, would assume that the rate of change is constant across all ethnic groups. So I'm already saying that these branches might mutate faster. Well, how fast would these branches mutate if they're this long? Perhaps super fast. Or perhaps the DNA is degraded. Whatever it is, the way these lineages fall on the mitochondrial DNA tree is the basis for me questioning the utility of these sequences or their utility to argue against the young Earth model. The point is, this paper addressed some of the objections that had been raised and continued and advanced these predictions I had put in print. Okay, so just to stop here and summarize, by 2015, we had evidence that the rate of change of mitochondrial DNA was consistent with the young Earth timescale. We saw that the structure of the mitochondrial DNA tree was consistent with the biblical anthropology. We had some indirect evidence against ancient DNA, not very strong evidence, but some of it was there. And some indirect evidence to say that the rate of change was constant. End of story, no. What defines science is the ability to make testable predictions. One of the things that is still untested is the rate of change in Africans, in African mitochondrial DNA. I said we know that the biblical explanation for the origin of the human species is true because it has produced a decades-long series of predictions and fulfillments. I'm stopping here just to reemphasize this point because I'm about to go on and give you some more predictions and fulfillments. One of the main arguments against mitochondrial DNA results at this stage, 2015 or so, and even to this day, if you look at the evolutionary literature, they'll say, it doesn't matter what mitochondrial DNA says or doesn't say. It's just a small fraction of your DNA. Remember, 99% of your DNA is this type of DNA, autosomal DNA. So who cares what the creationists say about mitochondrial DNA? They can't explain this. So in this case, just like mitochondrial DNA, DNA mistakes occur every so often, except it comes through both sides of your family tree. This is something I addressed explicitly in 2016, along with Jason Lyle helping with some of the math of this paper. I want to set up the argument this way. One of the main things that I showed in this paper is that there exists a contradiction between mitochondrial DNA clocks and nuclear or autosomal DNA clocks. If you measure mother-daughter mutation rates for mitochondrial DNA, it's fairly fast. You can dial that mitochondrial DNA clock back to 0 in 6,000 years. If you measure mom's autosomal DNA and dad's as well, compared to the daughter's autosomal DNA, and from that perspective, the rate of change is kind of slow. It's 80 mutations per generation, but with a data set of millions of DNA differences even in this room, you can't dial that clock back to 0 with mutations in 6,000 years. So that's what this bar represents. In 6,000 years, you should produce about roughly 40 DNA differences in the mitochondrial DNA. That's approximately what you find among non-Africans. If you look at autosomal DNA, there's millions of DNA differences between us on average. And in 6,000 years, 80 mistakes per generation, you produce just a handful. So there's a contradiction. There's a contradiction, though, for young Earth creation and for evolution. So what do I mean? Well, what if you don't have 6,000 years, but you have millions of years? That'll bring this bar way up here. And now you can capture what you see. Evolution will say, hey, we can explain millions of autosomal DNA differences in millions of years. The problem for evolution is this side. In millions of years, this bar will go shooting up this way through the screen. Way too many differences in millions of years. So they've got a contradiction just the opposite way. Young Earth creationists, how do you explain young Earth creationists can explain mitochondrial DNA. There's a contradiction with autosomal DNA. The evolutionists say we can explain autosomal DNA, but we have trouble with mitochondrial DNA. So how do you resolve that contradiction? I think the evolutionary model has a tougher time of it. Remember, I said earlier that one of their objections was, the creationists haven't considered natural selection. That's surely what they invoke here. Natural selection takes this huge number of differences and reduces it. Okay. Well, what role does natural selection play over on this side of the screen? Or is it just something we invoke willy-nilly whenever we're going to make the numbers work? That's what the evolutionists have accused the creationists of for years. They say that we say God did it, and this is our rescuing device. We don't have an explanation. God did it. That's pseudoscience, that's religious, they say. If the evolutionists aren't careful, natural selection will become God did it for them. Natural selection did it. Well, how? What predictions does it make? It did it. That's not science. The explanation I advanced in this paper, so I'm going to skip that slide. It just means that there are other species in which we saw the same thing. This is the point I've been trying to advance here, that the autosomal DNA that's from both parents, biparental, and mitochondrial DNA from one parent, mom, those clocks contradict each other. And that was true for both young Earth creation and for evolution. Evolution, I think, will have trouble explaining this. I'm speeding up here just for sake of time. What I think explains this contradiction and resolves it very well for young Earth creation is a recognition that these types of DNA are inherited in different ways, back to the basic genetics that I referred to again a few slides ago. Mitochondrial DNA comes only from mom. So what? Well, what happens in the beginning? God creates Adam and Eve. Adam has mitochondrial DNA, Eve has mitochondrial DNA, but only moms pass it on. Was Adam's mitochondrial DNA different from Eve's? Could have been, but it's not relevant as an explanation because he doesn't pass it on. Only Eve passes it on, so effectively, creationists, in a sense, are forced to explain all mitochondrial DNA differences by mutation because Adam can't contribute to that pool of diversity. You could, I suppose, say that God created Eve with eggs of different mitochondrial DNA sequences, but that just seems a little bit of a stretch to me. What about autosomal DNA? This is inherited differently, comes from both parents, not from one. I like to visualize autosomal DNA in this way. This is a microscopic version, is staining of it's called a karyotype, but your chromosomes. Humans have 46 chromosomes. There are 23 pairs. Pairs, as you can see here, for every long chromosome, they kind of look like wet noodles. For every long wet noodle, there's a corresponding wet noodle of a similar size. So if there's a really long one, there's another long one. If there's a really short one, there's a really short one. With the one exception being down here, this is an X chromosome, this is a Y chromosome, they're different sizes. This is a male. For ladies, it would be this longer one paired with another longer one. They come in pairs because one member of each pair comes from mom, and the corresponding member comes from dad. And just to clarify, it doesn't mean that dad codes for, let's say, the right hand and mom for the left. Both code for your entire body in ways we still don't fully appreciate or understand. The interaction between those two produces what you and I look like. That's why you look like a mix of your parents, because you have information from both. That's how my DNA would look. I've got a version from mom, a version from dad. I look like a mix of my parents. What about Adam and Eve? No parents. This relates a little bit to the question of, did God create them mature? Is God being deceptive if he creates Adam and Eve with genetic diversity? I'd say no, because he tells us exactly what he did. He created them. He could have created them with genetic diversity, or without it. I think it makes sense for him to do it. So let me actually, for sake of time, make a very brief point here. I think I can advance a theological argument that God did not create Adam this way. So if God creates Adam with no differences between his two versions of DNA, and then he makes Eve from Adam's side, and she has no differences between her two versions of DNA, so that Adam and Eve are basically genetically identical. When they fulfill the command to be fruitful and multiply, they're effectively reproducing by cloning. That just strikes me as weird. I also think there's good scientific arguments to say that Adam was likely created this way. Evolution attributes all differences to mutations. So they basically say their version of Adam, hundreds of thousands, millions of years ago, would have had very little genetic diversity and would have mutated it over time. Again, I'm saying Eve would likely have been created from a side, also have different versions of her DNA, as if she had parents, so that when she and Adam reproduced, they could produce a diversity of offspring in a single generation. In other words, my explanation for why the mitochondrial clocks contradict the autosomal DNA clocks is because they're inherited differently. One has the possibility, I should say this has the possibility of being explained by created DNA differences. The other one is inherited in such a way that you can't invoke that. It's all mutations. I think that makes good sense. It also makes testable predictions. So the point of bringing all that up, even if you didn't follow all the details, was that when we brought in a new DNA compartment that ended up at first glance seeming to contradict the earlier results, the resolution actually ended up confirming the original mitochondrial DNA results, and it led to additional predictions. It has to do with DNA function and such. Again, for sake of time, I won't go through it in detail. Okay. Now we're 2015. A lot of this is summarized in this book, Replacing Darwin. Now, I'm just about out of time, and I'm going to race through, or summarize, I should say, not race through. I'm going to summarize.
Don't race.
Okay. I'll summarize here the highlights of what's been a spectacular 5 years of research into human origins that's led to this latest book, Trace. So in a nutshell, and you can find us, I'll be giving a longer version of this in this ongoing video series we have through Answers in Genesis YouTube channel. We're up now to, I think, video 8. I've been sort of summarizing the chapters, and then I'll have about 5 videos. We've already pre-recorded them that go through some of the more of the history, the backstory to how we got to the Y chromosome, the male inherited DNA, working so well. So just to stop for a moment and review the biology. When I say Y chromosome, I'm talking about the male inherited DNA, only in males, inherited only through males. This project got started about 2017, not in this book, but with Rob Carter. I was working on mitochondrial DNA. He was working on male inherited Y chromosome DNA. At that time, I said, okay, we've got good evidence for there being mitochondrial DNA clocks going back 6,000 years, time element. We've got within the mitochondrial DNA tree, what seems to be an echo of the biblical anthropology of the flood, 3 ladies there at the beginning. The next biblical event seems to be Babel. And at that point, I initiated a collaboration with a linguist, PhD linguist, who was at one point associated with Wycliffe Bible translators, to see if we could overlay the genetic map of humanity based on mitochondrial DNA with the linguistic map of humanity. Babel is fundamentally a linguistic event. God confuses the languages, but that linguistic event is going to have profound genetic consequences. Long story short, as you might expect, sort of in retrospect, it was sort of a, well, duh moment for us when we finally realized it. You can change your language, you can't change your DNA. That's really obvious. But there are positions of overlap when there's a people group, let's say New Guinea, that has been isolated for thousands of years. There they have linguistic isolation, they have genetic isolation. Long story short, what I ended up doing is basically reversing my approach. Instead of saying, okay, let's go creation, fall, flood, Babel, and look for the echo of Babel, let's fast forward to the present and work our way backwards to Babel. So the biblical record doesn't stop with Babel. It then gives us the history of Israel and such, Old Testament. There's all sorts of migration, conquest, intermixing. There's 4,000 years of this, biblically and extra biblically, after Babel. So why don't we start here in the present and work our way back in time, unraveling all the genetic echoes of extra biblical history until we can get back to the beginning. And so the first extra biblical historical event we tried to analyze was the ugly and sad history of the transatlantic slave trade. Why? Because we had DNA in the public databases from Africans and African Americans. You can also find very detailed records of the exact number of slaves that were kidnapped in Africa. How many made it over to the Americas? How many disembarked in Brazil? How many disembarked in the Caribbean? How many disembarked in North America? So you've got this great genetic data set, this great historical data set, compare the two and find out which time scale makes the best sense of that. When did the African branches and African-American branches diverge? We almost got to the point of publishing this. We presented this at the Creation Research Society meeting, maybe 2017. It was, I forget which, this was Ann Arbor. Yes. Is where we presented this. Got to the point of presenting, and then I always play the devil's advocate when I write a paper, saying, what are the critics going to say? What are the reviewers of this paper going to say? And the thing I couldn't shake was they're going to look at our results and say, hey, great, looks like the young Earth model makes the best sense of the data surrounding the transatlantic slave trade. But what about the rest of the tree? How do you explain that in Europe? And this event over here, and this event over here? There's just all these other questions about the tree that I didn't know how to answer. Long story short, that sent me chasing for about 2 years, a whole bunch of other data. I ended up working almost exclusively on the Y chromosome data and published 2 papers at the end of 2019. I'll show you the titles here in a moment. Trying to find the echoes of the history of civilization in Y chromosome DNA. Well, it turns out we also found that the father-son mutation rate, so again, just like mitochondrial DNA, there's mutations that occur from generation to generation. In this case, it's about 3 mistakes per generation. Every generation, there's new mutations that are laid down. And this also means there's new branches of the family tree that are laid down. And the first 2 papers that I looked at and discussed the evidence in the evolutionary literature for the father-son mutation rate, and also looked at the history of population growth. That's sort of a technical point, which we can talk about another time, but 2 very strong evidences, not only that that a third genetic compartment fit the young Earth time scale, but that the rate of change was indeed constant throughout human history. So this second paper right here, where I looked at the history of population growth, is one of the strongest arguments right now that this rate of change has been constant. As I was answering and explaining this to my evolutionary critic 3 weeks ago, this is when he was typing and moving his mug around and not listening. And you can still find him producing even more videos saying Jeanson didn't answer this question. I mean, you can't make this stuff up. We fold our hands, say, hallelujah, we're done. No, science keeps making testable predictions. What these results predicted, and so here's why I'm going to summarize a whole rapid fire series of discoveries that have been made. Again, the point being there's a history here that fulfills what the evolutionists have demanded. If the data match globally, you would expect to find regional matches, and you can find 2020, another paper I published on the history of Native Americans, Native American population growth and decline, that you find a clear echo of in the Y chromosome tree. There's yet another example of this, 2022, in this book that I just published, Trace. Of course, as I just mentioned, the driving motivation behind all this was We had made these observations for the transatlantic slave trade. Well, what about the rest of the tree? Do you see the history of civilization, the Mongol Empire, the Russian Empire, the Roman Empire, the Greek Empire? Do you see those echoes in our DNA? And that's essentially what the book tries to show. Yes, indeed, you do. And you only see it when you have the young Earth time scale. When you take the human family tree and you stretch it out over hundreds of thousands of years, these echoes disappear. I can also tell you that along the way, we found the genealogical echo at the base of the Y chromosome tree of the Genesis 10 genealogies. You can find Noah, Shem, Ham, Japheth, even some of the genealogical differences, Genesis 10, Shem has a longer genealogy than Ham does. You see that level of detail now in this Y chromosome tree. And this is literally a fulfilled prediction. You can look at the 2019 paper where I looked at the history of human population growth, and I say, I don't know where Noah is. He could be here, he could be here, he could be here, but not here, because the data doesn't work here, but it does work here, here, and here. And this didn't come together until April 2021. Perhaps what makes this whole sequence of events so powerful is that it creates a strong explanatory predictive framework for doing research in the future. So, Lord willing, I'll be working on human history and the genetics of human history for the rest of my career, uncovering the history of every people group on Earth. That's the project going forward. And this framework of matches is what gives me the ability to say, if I want to find the ancient Assyrian lineage, we need to go to this part of the globe and search at this depth. I need a sample size of this many men. It's an extremely powerful framework that says, I can predict for you where to go looking. And the reason I phrase it that way is because this is what Neil Shubin makes a big deal of in his book, Your Inner Fish. He says the reason evolution is such a great idea is because we've got an evolutionary sequence of events in the fossil record. And if I want to find a fish to land transition, I've got to go look at these layers in this part of the globe, and voila, we found it. That's his logic. This is why evolution is so believable. And I'm saying now we've got in creationist genetics a version of that, if not better. We want to find the ancient Assyrian lineage. We want to find the Sumerian lineage. We got to go looking here in this part of the globe and sample this many men. I don't think the evolutionists can do that. They don't even expect to find these sorts of ancient branches that I'm predicting. That's what makes this so powerful. So just so multiple independent lines of evidence are confirming the Y chromosome history. There's now female inherited DNA, biparentally inherited DNA, male inherited DNA, all telling the same story together. And it's the Y chromosome data that's producing even more fruit going forward, the main tool I'm using going forward to uncover human history. So here's the father-son mutation rate paper that I referred to just a few minutes ago. This is the human history of human population growth at a global scale paper. This paper talks about the history of the Native Americans. Just to give away some of the punch here, we can see genetically that today's Native Americans were not the first. There have been multiple settlings of the Americas before Columbus ever arrived. It's pretty remarkable. So this book, and I could get in trouble for saying this, in a sense is not primarily an apologetic book. It's first and foremost a history book. Why? Because creation science has now advanced to such a stage where we can just do science. This just does history. Because we can, and it's doing it better than the evolutionary community is. So the fact that such a book can be written where we're just reading off history from DNA, and there's some apologetics in it. Here's the history of measuring the father-son mutation rate and so on. But simply to read off history, that is the apologetic argument. You shouldn't be able to do this if the young Earth model is pseudoscientific. Now, if you want to participate in future research, and again, this is 2022, it's part of a sequence of predictions, fulfillments. If you want to participate in future research, you can go to the Answers in Genesis homepage, answersingenesis.org/go/traced. It'll take you to some of the supplemental material. We're going to update this page here, hopefully in a little while, but you should find a button right here, Hidden History of Every People Project. You can click on it or just scroll down, find a place to enter your name, email, message. We've gotten, I'd say, close to 600 people responding this way. So in some cases, it's people who've taken genetic testing results. Some people say, well, I want to participate in future research. I've had international people. We've got a Native American study group that's formed because I have 2 Navajos, Yupik from Alaska, a Cherokee. There's an Ojibwe lady as well. They're believers, creationists. They say, we've never heard this. We want to find out our own history. How do we do this? So we form a study group. There's an East Asian study group. Hmong lady, 2 Koreans, and also Karen from Burma. We want to find out history. Okay, let's get together as a study group, and so on. South Asians. We want to dig in more into the deeper history of India. Who were the people of the Indus Valley civilization? There's all sorts of things that are springing up nationally and internationally. So if you want to participate or you've got a genetic testing result, how does this connect back to NOAA? Fill in that form, it will straight to my email. I can correspond with you that way. So the point of this talk was to show that there are more answers to the origin of species than we commonly assume. And the point I wanted you to see here specifically is that on the question of human origins and human history, we've got multiple independent lines of evidence pointing towards the veracity of the young Earth model. But more importantly, is the timing of how this has come about. It's not just, hey, we glom together a bunch of evidence retrospectively. No, we put our necks on the line. We started with the data that we had with mitochondrial DNA. It worked. We made predictions. They worked some more, made more predictions in a different compartment. Those worked some more. Those made more predictions. Now over to Y chromosome, more predictions, and it's ongoing. That's the standard the evolutionists have demanded that creationists are now meeting. And the way I end the book, Traced, is basically to say, hey, don't believe me? Let's see how it continues. That's the gold standard of science. That's what evolutionists have demanded from creationists for 40 years. We're now meeting that. That is a game changer. That's an answer. And I'm saying that I'm optimistic then that this progression for the question of human origins will find an echo in the rest of the species, because just a few months after I published this book, which made predictions on genetic change, which made predictions on the rate at which species would form, lo and behold, January, so this is October 2017, January 2018, Darwin's finches are announced in a major paper in Science magazine. So the icons of evolution, textbook examples of natural selection and action found in these islands just off the coast of South America. Peter and Rosemary Grant's group announced that they had observed in real time the formation of a new species. There's the paper. And I didn't actually find out about it in January. I found out about it in February when the evolutionary community was celebrating Darwin Day by saying, hee, look what we just discovered, let's rub this in the face of the creationists. Turns out if you crunch the numbers for this paper and say, okay, here's the rate of change within Darwin's finches, the rate at which new species are forming. And they also talked about the mechanism by which they form. If you take all those numbers, compare them to what I predicted, you find an almost exact match. I don't have time to go through all these numbers, but it was one of the best gifts to replacing Darwin that I could have asked for. The textbook icon of evolution, forming a new species, but at a rate and in a manner that exactly fits the young Earth model. You can go to our website to find these articles. They're not in the book Replacing Darwin because they fulfilled predictions that were made in Replacing Darwin, and we found out the answers after the fact. So I'd like to think this is a marker of things to come. Again, human origins being a marker of things to come. This book, just again, briefly to review here, is the broader origin of species question. I wrote this with skeptical audience in mind. 10 chapters of science, closes with the gospel, assumes the reader has objections to creation science or just assumes evolution is true for X, Y, and Z, and we walk through that evidence and we make testable predictions. This book, again, is the simpler version of it. Cliff Notes has 2 parts. First part, several short chapters. This is really more like a booklet. Here's how species form. Here's the answer to the origin of species. Part 2 is here's how you engage an unbeliever or an evolutionist with these data. Here's what they're going to say. Here's the counter example to it. Here's the counter argument.
What's the best way to get that book? Because I tried looking at Amazon, it's not on there. The other book is, but not the simple.
Yes, good question. I think this is on Amazon. New Leaf published this. This, I think, is in the no man's zone of being between Ancestry and Genesis and New Leaf Press. So we published this initially. I think New Leaf Press bought the rights to it, and so now it's hanging out in no man's land, not on Amazon. I know we have it in our warehouse because I filled orders for this when I put some time in there. So I'd say probably go to the Ancestry and Genesis web store, so ancestryandgenesis.org, and just click on store. I think you'll find it. I know we still have stuff in stock. I don't know at what point New Leaf will begin printing it.
Quantity discounts like for Bible class, or it's just...
My guess is probably yes. So if you call into customer service, I don't know the answer offhand. I'm one who's done bulk discounts myself. So when I graduated college, I bought a whole case of Refuting Evolution 2. Actually, I think I asked about bulk discounts and I wanted to buy 10 copies or something. She said, well, this is the price. And she said, but if you buy a case, it's this price. I said, I'll get a case. And she said, well, no. So no, I want to give her away. Give me a case. And so I got the case discount. My guess is yes, if you buy them in quantity, but I don't know offhand. I probably shouldn't speak for the business office, but you can tell me to talk to me or something. Maybe I'll give you a discount. 1 hour summary version of this you can find on DVD or on our streaming service, answers.tv. This is the new book that just came out. Again, it's primarily a history book where where creationists are taking the lead in this field of historical science, investigations of history, making predictions for the future, explaining what we see. That's the major focus of this. But again, assuming that this is going to find its way into the hands of unbelievers, so it ends with the gospel explained in a sort of in a historically sensitive way in light of what we all just read. Here's what it means for you and me. So there's, oh yes, there you go. Yes, this also has it really, half this book is actually, you can see it there, is images and figures, about 235 color plates, because history for me is just so visual, to see the maps and the correlations between this genetic branch and the Muslim Empire. They overlap, that sort of thing. Great visual tool to work through it that way. We have the Museum and Ark Encounter, of course. Come visit us in Northern Kentucky. If you don't get a chance to ask your question today, I'm at 2 minutes from a break. You can find me online. Again, I gave you the way to contact me directly through email if you want to participate in future research, or if you just have regular questions, you can find me on these social media platforms as well. I'm on Facebook. I don't know for how much longer. I don't have Truth Social listed here, but it'll be the same handle as this, at Nathaniel Jeanson. I'm going to stop here and do maybe a minute or two of questions. And then
Daniel, I've cut my presentation down, so you go ahead and take questions for 10, 12 minutes.
okay. And these look like these were, I'm looking at the Q&A. I'm looking for you. I'm looking at you all online. So if you have a question, type it in the Q&A.
There was a question earlier. Can I start off with that one?
Yeah.
There? It related the last time you mentioned it this time. How does evolution explain simultaneous origin of male and female over a wide variety of times?
Yeah, so the question being, how does evolution explain the origin of male and female multiple independent times? And I think the answer is, I don't remember the classification offhand. I think first of all, they would do their best to explain it one time. They don't want to have to independently explain, invoke miracles multiple times to explain sexual reproduction in this branch, and this branch, and this branch, and this branch. They would likely want to bring all the branches together, explain the origin one time, and then just have it inherited over multiple lineages. Now, how do they even explain it the first time? How do they get female and male to appear naturalistically simultaneously in evolutionary history? They can't apart from a miracle. And I say that because I've watched the responses to Michael Behe's work over 20 years. And the evolutionists tried to critique it. And I mean, you can categorize how the evolutionists have responded to it. They either change the subject, they misdefine things, they use irrelevant examples. None of them can grasp with the details of what he said and give a step-by-step explanation for it. So even if they reduce it to one event of explaining sexual reproduction one time, they're still at a loss. Darwin's test of evolution, the means by which to refute evolution, occurs. It's effectively, what does he say? If it can be demonstrated, any complex organ existed that cannot be explained by numerous successive polite modifications, his theory would absolutely break down. So it breaks down when you try to explain the origin of male and female. Other questions? And again, I'm looking at the Q&A here if you're online, so it should pop up here, and I'll answer those. Go ahead.
The question was when you were showing the slides with the cylinders and you all had a little bar going on through the top of it. Where's
Yeah.
the bar with?
Click on. Someone raised a hand. Tom and Beverly raised a hand. If you can type your question in the Q&A. I'm speaking to the online folks here. There we go. Screen sharing is stopped. Well, it'll resume here in a moment. As soon as I go back to my bar slide, and then we can talk about the same thing at the same time. And now I got to do screen share.
Yes.
Now we got it up. Ask the question again.
Question is that fire going through the top of the cylinder there?
Oh, yes,
Fire-
the black line right here.
Because that one on the kilo one is so much longer than the other ones.
Yes, good question. I apologize for not explaining this the first time. The black bar represents the statistical range, essentially. I think it's the 95% confidence interval. I have to go back and check my own paper to remember exactly, but there's inherent variability in the measurements. So, I mean, biological data is messy. So when you say how many mutations occur per generation, it's 5 plus or minus 2, something like that. There's also inherent, what I also try to do in these studies is account for the variation in generation time. So for humans, some moms are moms in their teens. My mother had my sister at age 43. So there's a wide range of when humans reproduce. I think the fruit fly generation time is temperature dependent. So there's a range at which they can go from fertilized egg to sexually mature adult, how long that generation time is. That's part of the reason there's variability, because I calculated to make sure we're comprehensive for the very short generation times, for the very long generation times, because that's something the evolutionists could invoke. Well, your numbers only work if you assume a very fast generation time. If you slow it down, now we can explain it. So all that's sort of built into these long black bars. The asymmetry here, I have to remember exactly what it comes from. I tried to put because it's such a big range right here, I explicitly called out here the lower end. The lower end is still almost 100,000 mistakes when the actual number of differences among these fruit fly species is maximally 2,200. So even the lower end of that is way higher than anything that we actually see in nature. But the black bars there are to account for all of that variability in the measurement and the ways of calculating it as well.
So that's the confidence interval.
Essentially, yes. Other questions? Yes.
In one of those brief online videos you had, it was about the Aztecs and the Olmecs being in the Americas, 1,500 BC.
Yeah.
That you had traced back that far. And then also that prior to that time, there's also a possibility of people being there. But question is, if you're able to, or in your ongoing research, you would be exploring tracing back further to before 1500 BC of other people being in the Americas?
Yes. So just for the online group, the question refers to this video I'd done in our ongoing series. Also chapter 12 of the book, Where the Native Americans Come From. And so you can get the 30-minute version of this. Related to something I'd alluded to earlier, if you look at the dominant Y chromosome lineages in the Americas today, they branch off from Central Asia 1,000 years ago, 1,700 years ago maximally. So the oldest branch we have on record comes across the Bering Strait about the 300s to 600s AD. And of course, archaeologically, the Mayans go back to 400, if not 1,000 BC, and then the Olmecs, the cradle of civilization, all the way back to 1,500 BC. So who did these people come from? Or let me back up a second. So it looks like whoever is here right now replaced or wiped out the Mayans and the Olmecs. And it raises the question, who did those people come from? Can we detect those lineages? And my hope is, yes, that's part of the goal of this Native American study group, is can we detect among living Native Americans something more ancient than what we see right now? It may be a needle in a haystack, it may be extinguished. So part of the challenge in this is, even in mainstream science, the current estimates for population growth and collapse in the Americas is that there were, so middle of the road estimate would be 50 to 60 million people here in the Americas in 1491. That's almost the population of Europe in 1400. Then about 80-90% of them disappeared because of diseases, enslavement, other tragedies. And that happened for about 300 years, and then about the 1800s, the population finally recovered. So after 90% of the population disappears, and what's left is still these two lineages that have arrived late, could there still be preserved something more ancient? Or would those have been lost in this massive population collapse? And that wasn't the only population collapse. Before Europeans arrived, the Mayans themselves, 750 AD about to about 900, is the Mayan collapse, where the city's empty, the rulers stop inscribing their histories in the pyramids and such. Pyramid building basically stops in the lowland Mayan area. Nobody knows. The mainstream scientific community has as many explanations for what caused that collapse as they have for the extinction of the dinosaurs. So it's highly debated. So you've got, and if you go back even further in history, you've got the Olmecs who are going into decline as the Mayans rise. So is there an Olmec lineage that has survived? It would have to survive their own decline, then the Mayan rise and Mayan decline, and then the whole Native American rise and then massive decline. And I think the chances are probably low, unfortunately, though that's not going to stop us from looking. I think there was a hand raised or a queue. Still, okay, these are older ones. I saw a hand raised, but I didn't see a typed question from online. Anyone locally here? Questions? Yes.
I mentioned last night briefly, what I see as the primary secular attempt to answer what you're answering, and that's
Yeah.
the work of David Reich, as
Yeah.
well as, if I look on YouTube, I see Razib Khan's name all over the place, also addressing the same things you're trying to address. Now, I got traced just like last week because of the back ordering issues, so I haven't had a chance to read it yet, but therefore, it's more of an open-ended question. Do you have a reaction to the type of work that David Reich and Razib Khan and maybe others I don't know about
Yeah.
are doing, and how it compares and contrasts to what you're doing?
Yes. So the main objection here, and to repeat the question for the online audience, the question is essentially, what do I do with ancient DNA? Some of the main arguments, some of the main research being done by the mainstream community right now in human history deals with getting DNA from fossils or from graves or from so-called Stone Age settlements and so on and so forth. David Reich is the leader of this. They've published all sorts of, not just Neanderthal sequences, Denisovan sequences, but all sorts of burials. And those sorts of data in which they've got a radiometric date in a DNA sequence would be a natural contradiction, what I'm doing here. And this is what's been raised. There's been about 3 PhD biologists who have responded to TRACE, already published reviews and criticisms of it. What's somewhat entertaining to me, so I've put in print, going back to sort of where we started at the beginning of this talk, I've put in print my explanation for why the mainstream community rejects creation science. I say because they don't read our literature. Here's 3 examples of people who say they've read our literature. They all basically are tee-heeing, saying, we've got him now, he's totally ignored Neanderthal DNA and fossil DNA. I explicitly addressed this 2.5 years ago in this paper, which none of them seem to have read. And when this guy came online from Rutgers 3 weeks ago and said he read this, and I asked him, you can watch the hour debate that I did with him. Have you read this paper? Yes. Well, can you tell me what my main point is? No. These sorts of things. Okay, well, I struggle to understand how you can say you've read this paper and can't articulate the basic points of it. In this paper, so my argument for why I think this sort of DNA from the deceased is unreliable is essentially, there's 2 parts here. This is connected online, right?
Yes.
So I can go look for the paper. Because I want to show you a graph. Chromosome clock, Jeanson. It's a dual part. Here we go. I have to change my screen sharing.
Correct.
I think I got it now.
Yes.
Okay. So here's where I'm testing a bunch of aspects of my model all at once. This paper tests where is Noah. This paper tests ancient DNA. It tests whether or not the mutation rate exists long term. I think I have a graph in this paper. So let me just tee up a few things here very quickly. The history of human population growth has been estimated from archaeology and historical records. So the Roman Empire, the Han Chinese Empire did censuses of their people. We have those data saying here's how many people were under the Roman rule. That's a historical time point. You can go to archaeological sites and say, okay, here's this monumental structure. Here's the amount of people you would need to supply this labor force and to feed this labor force. You can make estimates from archaeology as to how many people were in existence at certain points in history. These black lines right here represent those mainstream estimates independent of genetics. Comes from a book that was written in the 1970s before we had any genetic data. These blue lines represent the reconstruction of human population history from family trees. So the It's a technical point, but in a sense, it's also a kindergarten point. If you draw my family tree, me and my wife, and then 4 branches going down to our 4 kids. Branches represent population size. The more branches, the larger the population size. Fewer branches, the smaller the population size. It's pretty straightforward. So what I did then is say, okay, 3 mutations per generation between fathers and sons. Let's assume it's been the same. That means you've got new branches being laid down every generation. We should be able to just count the branches throughout the human DNA tree and reconstruct human population history. That's sort of the method in a nutshell. And I can say, well, if we put Noah here, here's the shape of the population growth curve. Or we put Noah here, the shape of the population growth curve. If we throw in Neanderthal DNA, shape of the population growth curve. That's what I'm doing here. These black lines are the test data set. If my methods are working, it should accurately reproduce the history of human population growth. This graph right here, this blue line, you might be able to see it here on the screen. You might need to zoom in if you're online. You can see here that this blue line misses the black line until about the 1200s. Then it rises up and meets it. This is taking DNA just from living men and putting Noah in the position that the evolutionists say is the start of human history. It's a poor match. The percent match is at 27%. So the match between the blue line, the blue line captures 27% of history of human population growth. Then I picked, then I throw in Neanderthal DNA. What if you take Neanderthal sequences, throw them in the tree, reorient the tree the evolutionists want, the match drops even more so. Between this blue line, the black line, the match is only 14%. What if you leave out Neanderthals? And this is where I want to go. What if you leave out Neanderthal DNA, any DNA from the deceased, only take DNA from living men and put Noah in a position that actually turns out to be where I think he is, reconstruct population growth, you get a 95% match. These blue lines and these black lines, very close overlap, gets this hockey stick shape. Again, these 2 black lines are because there's a range of estimates for human population growth. The 2 blue lines because there's inherent statistical uncertainty, and so this is the range of uncertainty. So the point is, when you take the DNA from living people, you assume a constant rate of change. You've got the young Earth time scale, all these various assumptions. You get a tremendous match to the test data set. This is not something any of the critics have dealt with. They've actually assumed the opposite. They've ignored this paper. Joel Duff is a professor in Ohio, biology professor, who has basically 2 points of criticism. And one of his points is Jeanson has ignored, or apparently doesn't seem to be aware of the existence of Neanderthal DNA. He seems to actually believe that I don't know what's going on, and I hope the problem goes away. Obviously, never having read this paper. So, and again, I was making this exact point, showing these graphs to the guy from Rutgers, and he's not listening as I'm telling him this answer, which addresses multiple aspects of his point. So this is my main argument for excluding Neanderthal DNA, because we can reconstruct human history much more accurately when we leave it out. What's going on with Neanderthal DNA? I don't know. Is it degraded? Is it contaminated? I don't know. What I can say is we have positive, strong evidence for a model that leaves it out, and it does much better by independent metrics than if we try to incorporate it in. So that's my main argument. Now, I should say here, so Rob Carter, I think he's got a critique coming out of Trace here sometime in the future. I've been encouraging him and others saying, if you hold to ancient DNA, develop a model. This is healthy for the young Earth creation science movement. 99% of today's men have yet to take a Y chromosome sequence test. So we're at the early stages. I would argue that the young Earth model, and this particular one I'm advancing, is the one that's working better than anything else. Science is pragmatic. It's working, you chase it. But at some point it might stop. And so at that point, it'd be nice if we had an alternative model to go chasing. My hope is it doesn't stop and just keeps on working. But let's say that day comes, it'd be nice if we had another young Earth creation model that explained the data even better. We can fall back on that. So I think this sort of multiple ideas is healthy for the young Earth creation science movement, a sign of its maturity. For the time being, I'm chasing this because it's working. And that's the gold standard of science from my view. Now I see there's a Q&A question up here from one of the online folks. Okay.
Last question.
Or we can skip it for sake of time. Okay. So John says, won't pretend to be well educated on the subject. Did actually watch a video from Daniel Cardinal. That was the Rutgers guy I was just talking about a while back, where he said that you were using the mutation rate as a substitution rate when it came to the mitochondrial mutation. Does that mean the evolutionists would expect the red bar to be considerably smaller based on how fast the population actually takes on these mitochondrial mutations and passes them on versus how many times they occur in a single generation? Long story short, what John is referring to is, yes, the evolutionists recognize that mitochondrial mutation rates, in this case, they probably say the Y chromosome mutation rates as well, are way too fast for evolution, the evolutionary timescale to work. So how do you bring that number down? They would say, well, natural selection. So these terms here, mutation rate versus substitution rate. Mutation rate is the from father to son. Substitution rate is, well, how much does that get adjusted through long periods of time, where natural selection might play a role or other factors might play a role. So Cardinal is saying, I'm doing it incorrectly. And we're going to get to this in the video series that I referred to earlier as well, because it's a great example of they're responding to science predictions, not by proposing experiments, but by saying the book says so. Yes, what I'm doing contradicts what the textbook says, but science is built on questioning established dogma. And one of his point, and he's got videos out on this, saying he's doing it wrong because the textbook says so. I mean, we're supposed to be the ones with the holy book that you can't question. Apparently the evolutionists are as well. So does Daniel Cardinal say this? Yes, he does. Does he give any scientific predictions to back up what he's saying? Not really. So essentially what Cardinal is articulating is pseudoscience, where he says, you need to do it this way because the textbook says so. Okay, well, what predictions does that make? Predict for me the rate of change among African peoples. Predict for me the rate of change for grasshoppers, for mosquitoes, some species in which the mutation rate is yet to be measured. You won't find that. Okay, well, you've been very generous with your time. Thank you, John. Thank you all for your attention, and I'll turn it back over.